Roche, bonjour, hello, and welcome everyone.
In this video, we will learn about surveying biodiversity in water sources using an eDNA sampling kit. Environmental DNA, or eDNA, are tiny fragments of DNA that all animals shed as they move through the environment. It can be in the form of hair, mucus, skin, or urine. These tiny bits of DNA accumulate in water for a few hours to a few days. By sampling the water and analyzing the eDNA, we can see a snapshot of what animals are living in and around the area where the water was collected. This can be helpful for detecting if elusive species at risk are in an area, or if a harmful invasive species is moving into a new area. Each eDNA kit contains 1 pair of sterile gloves, 1 resealable bag with a small syringe and lock cap, 1 filter, 1 3.5 liter sample bag. Additional bags will be provided for subsamples, 1 specimen bag, and 1 data sheet. The first step in taking the eDNA sample is to put on the clean pair of sterile gloves. When taking a sample from a still water source such as a lake, take your sterile collection bag and lower it into the water at the shoreline of the lake. Once it is full of water, place this primary sample bag in a bucket. Using a new bag, repeat this process along the shoreline to take what are called subsamples. To combine your samples, carefully pour your subsamples into your primary sample bag and mix them together in the primary sample bag. Adding these subsamples to the primary sample helps to create a representative picture of the water source. For smaller water bodies, such as ponds, all the subsamples can be pooled into one kit. For larger bodies of water like lakes, collect subsamples within a maximum 400 meters distance along the shoreline. In rivers and streams, there is no need to collect or mix subsamples because the water is already getting mixed as it flows. To take the sample, place yourself downstream of the opening, open the bag, and let the water flow in. Once you have collected all your samples, they need to be filtered. Pull 50 milliliters of water from the sample using the syringe. Attach the filter to the syringe, screwing it clockwise until secure. Then, push the water through the syringe and filter. Repeat this process with more water from your sample bag, as many times as possible until you've either filtered all of the sample water or you can't push any more water through the filter. Next, detach the filter from the syringe. After doing so, fill the syringe with air. Once filled with air, reattach the filter and push the air through the syringe, letting the last remnants filter through. To make sure that the DNA in your filter stays stable long enough to get to the lab for analysis, your filter needs to be preserved using the preservative solution in the small syringe. Uncap the small syringe and turn it upwards so the tip is facing the sky. Then attach the syringe to the filter and push the substance through until a small bubble of the solution appears at the tip of the filter. At this point, cap the filter, turn it upside down, and push the rest of the preservative solution in. Once all the solution has been pushed through, remove the syringe and cap the other side of the filter. Fill out your online data sheet with the location, the date, time, sample ID, and amount of water you have pushed through the filter. Remember, your sample ID is the number that was assigned to you, followed by a dash and the number you've selected to represent the sampling location. When you're finished collecting and processing your samples, place the filter in the transport bag along with your completed data sheet and seal. The Nature Metrics team will analyze and sequence the data from your sample you've sent them and will produce a report about what's living in and around the water body.
Marcy.